I would like to introduce our first speaker, William Polvino, who is a doctor. He's the CEO of Bridge Medicines. He is a physician and pharmaceutical startup entrepreneur, having financed and run biopharma companies as CEO. Dr. Polvino is presently CEO and a board director at Bridge Medicines. Bridge is a pioneering drug discovery company focused on advancing promising early technologies from concept to clinic. Bridge was launched by Memorial Sloan Kettering Cancer Center, the Rockefeller University, Weill Cornell Medicine, and Takeda Pharmaceutical Company in partnership with Deerfield Management and Bay City Capital. It is a groundbreaking initiative that extends the work of the independent nonprofit Tri-Institutional Therapeutics Discovery Institute to advance innovative breakthrough science into clinically and commercially valuable drug products. TDI, for short, also a collaborative venture of the three research institutes in Takeda, is working on approximately 50 early-stage drug discovery projects spanning therapeutic areas that include infectious disease, oncology, neurology, and rare diseases. Bridge Medicine provides an unbroken, fully funded, and professionally staffed path from discovery to drug candidate. Dr. Polvino received his undergraduate degree in biology from Boston College, his medical degree from Rutgers Medical School, and trained in internal medicine at Massachusetts General Hospital. Bill?
Great. Thank you. It's a real pleasure to be invited here and to be asked to speak about two of my favorite topics, New York City and biotech. Biotech is a profession I love. I'm surrounded by folks who know real estate. I know nothing about real estate or designing a building, so this could be an interesting presentation. What I'd like to do in some brief time is talk to you a little bit about the obstacles that biotech faces. And I'd also like to tell you a bit more about the experiment that we have ongoing. I'm a scientist, so we talk about experiments on the Upper East Side at TDI and Bridge Medicines, as Nancy kindly explained. But before I do, I just want to give you 2 minutes about why I do this. Nancy did introduce me as a physician. I went to medical school. I had every intention of becoming a practicing clinician. I went to Mass General Hospital and trained in internal medicine, did the residency and all that. And something happened along the way at Mass General. In internal medicine, I found myself faced day in and day out with the ravages of diseases that had been progressing in patients for 20 or 30 years. And I found it enormously frustrating and disappointing that there was very, very little that I could do about that other than manage the decline, as people like to euphemistically say. I found that completely unacceptable. I absolutely do not accept the status quo. And as a result of that, I decided to dedicate myself to developing new drugs with the thought in mind that new drugs have a better chance of being able to treat more patients than I could as any clinician. So that's why I did this. You will find, as you go through the biotech industry, a lot of people with stories like mine. It's a very difficult industry, but it's also very, very important. When you think about what matters in life, this matters to me, and it matters to a lot of the people involved in it. Yes, we work for profit. Yes, we want to make it a successful business, but it's more than that. It's very important. We look at this as an opportunity to treat the children, our neighbors, our family down the road, and without continuing in this pursuit, I'm quite afraid that we can't advance beyond the point where we are. So we view it as very important. So I will talk a little bit about the obstacles and the obstacles that face biotech. You've heard many of them. I heard a wonderful presentation on innovation, and the speaker said, you know, there's a reason why an egg has a shell, and there's a reason why that's in a nest. It's very fragile. It's very difficult to do. And there's a lot of failure along the way. So it's somewhat of a special case in terms of attempting to innovate. What I really underestimated, including at the Bridge project, is how much inertia is present, and it's very insidious. Trying to break out of the status quo is a lot trickier than it sounds, and we will keep you posted in terms of our progress. I think it's wonderful so far, but the forces are afoot. And I'll tell you about Bridge, but I'm also going to say it's not the only solution. So I encourage all of those, and you'll hear from other speakers with different approaches who are in this fight to innovate. And we view it as an important mission. We're delighted to have the talents in this room helping, because there are a lot of aspects that go into it. First, let me begin with a slide that I borrowed from my good colleague, and it's wonderful. I get to work with David Scheinberg over at Memorial Sloan Kettering. This is his slide, and you can see Dante's Inferno down here, which he has lovingly named the Valley of Death. That's what happens to most good ideas. So over here, you have these extremely talented investigators and scientists who are coming up with novel cures for cancer, for autism, for Parkinson's disease. They have great ideas over here, but they can't make it over there to the human clinical trials. And there are a lot of reasons why. And there have been some initiatives put in place. Sloan Kettering, Rockefeller, Weill Cornell have some small investment funds that get things started. TDI took the next step, and Bridge is the step after that. So our mission is to find a way to take great ideas out of the laboratories and get them into those human clinical trials. I don't know how many of you have seen this slide. It's from KPMG when they wrote a report about the life science sector. Here's another interesting statistic. So what you see here on the left is the number of dollars in terms of NIH grant funding by geography in the United States. There's a city about 200 miles north of us that will remain nameless. That's top of the list. I'm a Yankees fan, so forgive me. They garner the largest number of NIH dollars. But right underneath that is New York. So New York is number 2 on that list. Very impressive in and of itself. What's even more impressive to me, and a bit frustrating, the next slide is the number of dollars in terms of private venture capital investing that went into these discoveries and breaking it out per NIH dollar, how much venture cap money went in. And if you're in Massachusetts, there was $1.32 invested for every dollar of NIH grant award. Here's New York at 6 cents, okay? That's unbelievable. And for me, I drifted into business. I look at a slide like that and say, wow, that's opportunity. And I really do think there's a tremendous amount of untapped potential here in the New York City area. So what is Bridge, and how do we approach this problem? We approached it by physically embedding on campus. So my office is in a Weill Cornell building, and those 3 universities occupy 2 blocks in the Upper East Side. And our mission is to get through that valley of death. And we can talk about all the things that you know, but I want to point out the dirty little secret in this industry, and that is financial failure probably accounts for more things never making it to clinical trials than anything else on that list. And nobody knows because nobody reports it. What happens to those discoveries is that they sit in the lab or they sit in the technology transfer office hoping to be adopted by somebody. But those adoptive parents never come along, and you never hear about it. So what we're doing is we're viewing it as there's a huge number of these projects that potentially could move forward, and we do it right on campus. Here's the slide of the kickoff meeting, and I think this is one of the key ingredients. I just learned moments ago that Carlo was in the room at the time. I was not, so I joined Bridge shortly after this slide. But one of the key ingredients here is you will see the 3 deans of the 3 institutions there. From left to right, that's Augustine Choi, Craig Thompson, and Rick Lifton. This doesn't happen without the backing from on top. And it's wonderful that I'm in a position where I can pick up the phone and get help from their offices and the people directly under them. That's Andy Plump in the center, who is at Takeda, and they provide a huge amount of support. They got this whole thing started with TDI 5 years ago and with Bridge 2 years ago. And then backing from, that's Carl Goldfisher from Bay City Capital and Jim Flynn from Deerfield. And to his, I guess that's his left shoulder, That's Mike Foley, who is the founding CEO at TDI, and some of you may know him. But that all got started about 2 years ago. And our mission's simple. We're looking for drugs that are breakthrough drugs that are clinically important and commercially valuable. And we're looking to do this by growing a vibrant collaborative culture, and that's really the secret sauce. If you've ever heard Mike Foley's presentation or his successor, Peter Meinke, they work in offices in the same building as mine. You heard from Nancy, over 50 projects. It's actually over 60 now. So I go into work like a kid in the candy store, and I'm looking at 60 potential drug products, and we pick and choose among them in terms of what's deserving to move forward. I would say of those 60, the number that would move forward without us, maybe one or two. So we're looking at the other 58 as a real opportunity here. And as you can see, it's got a lot of support from all three institutions. So here's a basic model. I like simple pictures, and we start with the Thomas Edison light bulb up there. So if you're an inventor and you're working at one of those institutions and you got a great idea, you can work with TDI and begin to create a molecule. And as that advances, it becomes a project inside this rectangular box. That's Bridge Medicines. That's us. So we sit there and we take that project in, and hopefully with success and some investment, we can spin that out as a new company. And what happens? Well, we advance the molecule. Sorry for the jargon. This is my world. We do pre-IND work. That stands for Investigational New Drug. That's the testing that you do on a molecule before you're allowed to do work in human patients. And then you submit that application, that's called an IND, to the Food and Drug Administration, and they give you permission to do clinical trials. So we are, if you will, a holding company. We take these projects in. We put little subsidiaries together, and with success and investment, we mature them so that they graduate into new companies. Pretty cool idea. It gets even cooler when you start thinking about that happening. And that's what we do. So our mission is to take in about 6 projects per year, ultimately hold 12, and be spinning out companies. And we are sort of in the early stages of that. So we're just bringing projects in, but over the next 3 to 5 years, we're looking to be spinning things out over time. And the model that's been set up to do this is an investment model whereby we expect to hold assets for about 18 to 24 months, mature them enough, spin them out, and we have the capacity to be up in the neighborhood of 25, 30 projects over time. There's a lot of excitement. It's great to have my job. I love it. And we'll see where this goes over time, but it's really wonderful to be talking to scientists who are doing cutting-edge research and talking to them about how to make a drug, how to make a company. I was in Boston, I was in San Francisco, and the entrepreneurial mentality needs to be more infectious here. It's getting there, it really is. But people need to be thinking about how to build a company. From day one, we're putting deep skill sets on a project. We're pre-financed. That's very unusual. So I don't have to go out and raise capital to get things done. I've done it. I've raised $300 million, and every dollar took me forever. I don't think anybody appreciates the fact that you can lose a year just trying to find money to get your company off the ground. The advantage at Bridge is we've got it in the bank. We're ready to start from the day that program moves in, and we're right on campus. So that's our mission in short. And as I say, it's not the only one, but our mission is really great from a number of perspectives, and we're looking to take breakthrough discoveries and turn them into drugs and new companies here. We view drug development as a relay race. Bridge is perfectly positioned to be running full tilt, so as that project comes in, we're already flying downstream with it. So it's wonderful to see you here. I know I'll be in a panel, I believe, so happy to field questions. So Nancy, I guess I'll turn this back to you.
Sure. Thank you. Thanks, Bill. So Bridge Medicine is one example of the life science kind of startup environment in New York City, dealing with the development of new therapeutics. And now we're going to kind of pivot a little bit and go to engineering biology, which is really about biology and design. And Dan Grishkin is the executive director of the Biodesign Challenge, which I actually helped him to launch about 3 years ago in 2015. And it's grown very, very rapidly. It is the world's first community biology lab. Well, this is GenSpace, which he also founded, the first community biology lab, a new model for science education and innovation. Biodesign Challenge is an international competition that pairs biologists with university design students to envision future applications of biotech. Dan has been a fellow at Data and Society, a research institute focused on social and cultural issues arising from data-centric technological development, the Woodrow Wilson International Center for Scholars, where he researched synthetic biology, and an emerging leader in biosecurity at Johns Hopkins Center of Health Security. As a journalist, he has reported on the intersection of biology, culture, and business for publications including Bloomberg Businessweek, Fast Company, Scientific American, Nature Biotech, and Popular Science. Dan?
Hi, everyone. Wow. It's good to be here. My name is Dan Grishkin. I am, as Nancy said, the executive director of the Biodesign Challenge. This is a little different, I think, for this crowd. I run a program where we partner university students with scientists to imagine the future of biotechnology. Now, we just heard a fantastic talk. Oh, wow, slides are back. So we just heard about the medical industry, but biotechnology is also at the agricultural industry as well as industrial biotechnology, and that's kind of, if you think about it as a pie chart, they're actually evenly dispersed. So there's lots of possibility for innovation in biotechnology that reach beyond just the clinic. We are partnering design students with scientists with the express purpose of exploring what is possible with biotechnology. And what we're finding is really impressive. Students who ordinarily have very little biotechnology background, partnering with scientists, are doing really fantastic work. And I'm very, very excited because one of our, actually the winner of the first Biodesign Challenge in 2016, is with us today, and they're going to talk a little bit about the company that formed. But actually, every year that we've been doing it, companies have emerged out of the Biodesign Challenge. So the students partner with the scientists. They're working with them throughout the semester. At the end of the semester, they come on stage at MoMA, present their projects before an esteemed group of judges, including Nancy, and then projects win, and then go on to either become, well, gallery shows or things that are exhibits in museums, or they end up going on to become companies. And so behind me, you'll see a couple of those. Alginet is one of them, and again, the team will be speaking in a minute. But also Royal College of Art had a team that ended up becoming a agricultural product that is now hiring. And then as well as more recently, a team from Colombia, the country, actually came up with a new material, which is a possible replacement for wool products. Lots of investment in just a short amount of time. Biodesign Challenge started in 9 universities as kind of an experiment, as a pilot. We've tripled the number of sizes of the number of universities that we've been working with. So 27 last year, we're probably going to go to 30, 35 this coming year. And then we started only in the United States. We're now in 9 different countries. So again, it's growing very quickly. And I really see this intersection of design and biotechnology as the way forward. Students, I think, often when you think about biotechnology, you think of scientists. I think scientists are part of that puzzle. They're thinking about what's possible technically. But there's a second part of that puzzle, which is what's possible socially, what's possible with the people who actually use that science. And I think that's where the designers come in and really think deeply about How do we use this stuff? What are we going to do with this science? How is it going to enter into people's daily lives? Big questions. And I think we're coming up with some really interesting solutions. So without further ado, I'm actually going to introduce the Alginet team. This is a picture of them winning the 2016 prize. That's our glass microbe. That prize gets handed down from team to team year after year. And really, Alginet and the crew showed what's possible, that you can actually take these ideas and bring them out of the lab relatively quickly and then turn them into companies. So Aaron and team, come on up. Grab a mic.
Cool. Thanks, Dan. So, hi everyone. My name's Aaron Nesser. I'm now co-founder and CEO of a company called Alginet, and it was, I guess, two years ago, almost 3 years, that we started with my co-founders Tessa and Alex over there, coming from this place, looking at the fashion industry, wanting to understand like why the materials we use to make all of our clothes just like don't work quite as well from an environmental standpoint as we want them to. So like thinking about recycling, where our textiles come from, and wondering if we could make a difference in that industry using biology. So we started as students coming up with this kind of concept, and after winning the Biodesign Challenge, developed it into something totally different. And that's called Alginet. So what we found, kind of surveying the whole world of bioplastics, biopolymers, how do we create plastics that don't come from oil? Plastics that, it's like insect shells, fish scales, and we found one called alginic acid that comes from one of the fastest growing organisms on Earth, and that's kelp, what you're looking at right there. That it grows in forests all over the world, but is also cultivated. Like 25 billion pounds or kilos a year are cultivated around the world. So it's this really plentiful resource, very fast growing, absorbs a lot of CO2, and we found a way to make a textile out of it. So we make a yarn. And it's, the whole concept here is, this is a non-toxic yarn. It comes from, living materials, and it's safe, so safe that you can even eat it. But that's right. You hear about things like ocean plastic pollution, microfiber pollution, how the kind of the effects of the fashion industry of clothing affect the earth, affect our oceans. And so we wanted to make something that if it gets out there into the ocean or like out there into the world, it's not going to do anything bad. So we've developed this over the last 3 years, working with some different research institutions, and have developed some product concepts, brought in some investment dollars. We have an announcement coming up next week that Dan will be able to pump that number up a little bit, so that's exciting. And I guess it's the interesting thing in our journey that I think is really relevant here is we came out of this space where we kind of had all the resources we needed coming out of a school, right? And then we graduated, and then there was nothing. No lab space. We couldn't even like set something up in the beginning. So there was like this period where we're like, oh my God, what do we do? So continuing to partner with institutions, we eventually got into an accelerator program that gave us lab access. But after coming back, going through that accelerator, we're back in New York, and we're like, where do we go for space? So please, if you want to talk about like kind of we're coming from this biotech with a little bit of light manufacturing. Please talk to us. We would love to tell you about like what we need. We'd love to learn like what you guys are thinking, and yeah, please talk to us. Thank you.
Yeah, why don't we do some questions? So, I mean, I think Aaron's story is actually really important because what we're finding with the Biodesign Challenge is that every year we're pumping out 2 to 3 to 4 small companies each year, all of them at the intersection of biotechnology, all of them at the intersection of biotechnology and design, and all of them are eventually going to be in a place like Aaron that are looking for small spaces to turn their startups into a reality. I think what makes New York special, and I should have mentioned this earlier, is that New York is the hub of design in probably in the world, certainly in the United States. And there's this really interesting thing that's happening where biotechnology is becoming a palette for these designers in a new way. And it's going to have to start here in New York just because we're all here melding. You're not going to find that in Boston. You're not going to find that in San Diego. And so there's real opportunity here. And I think that that's something that we should all be thinking about really deeply, because I think that's what makes New York really special in this ecosystem. So let's open up to questions. We have time for a couple. Yes?
Aaron, did you and your partners come from the fashion world, the science world? Where did you come from?
Sure, great question. We came from both. And so, like, the three of us are all designers. I also trained in biology, and we, work with material scientists. And so it's really that sort of, interdisciplinary approach that, like, we had scientists on one side, designers on the other, and we're passing back these ideas that turned into, materials and have now developed into, useful materials. Thanks.
So where do you go looking for capital to take it to the next step? Who do you approach, or who is out looking for you at what time to make that marriage?
Sure. One of the in terms of, looking at venture capital, there's a lot of interest and what we're really interested too is impact investing. And I think when we started raising money, that wasn't really on our radar. But we found, through Europe, in New York and in California, like a whole network of investors who are, have a little bit more patience, and are looking to do something good, along with make some money. So
As a part of their overall plan? Or
Yes.
both as a part of their overall
yes, and so there's a lot of interest from family offices as well. But I think for a material, materials are kind of like drugs. They can take a long time, and so it takes a investor with with patience. Yeah. Sure.
A couple of months ago, there was an article, I think it was either the Journal or the Times, the family that makes Legos, talked a lot about how to replace plastics that they do with other materials, as you're looking at. Mm-hmm. Are there hotspots that will attract money? What you're doing is replacing something that we all use on a regular basis, so that got some media attention recently. What you're doing is similar. Mm-hmm. Is that as we look at that 6 cents on the dollar that talked about a few minutes ago, is it science or is it money that will grow this industry? Do you run to the science or do you run to the money to find what to do?
Yeah, so I think it's very clear right now that there are lots of alternatives for the plastics that are petroleum-based. A lot of different options, some of which biodegrade, some of which come from waste streams, all of which are very interesting. The challenge with a lot of these products is that they're new on the market. Petroleum-based Plastics have been around for a long time. They're extremely inexpensive to make. And obviously they stay with us forever, which is a big problem. I think it's two things. One is regulation, right? So there needs to be ways to incentivize alternatives, and certainly in the plastics industry, and then investment. And I think these two things need to happen together. So is there technology out there? Certainly. Is there money out there? Yes. Is there a business plan that absolutely works, where we'll see an instant transition from petroleum-based plastics to something greener, bio-based plastics? I think that's going to take some time.
And if I could just add one more thing onto that, coming from our experience, one of the really key missing pieces is infrastructure. So all the manufacturing technology out there has been designed to, is optimized for what exists already. And so if you bring something different to a manufacturer, they're kind of like, ooh, I don't know if I want to get involved with that,
Right, because you're taking on risk essentially, but also, So I, as a reporter, I remember reporting on new materials, and the folks that were telling me the timelines on these new materials, it's about 10 years to go from the concept to an actual scalable plant for people to start buying these things and it finding its way into our clothes and our products. So it's time consuming. Yet I think at the same time, we're well on our way, and there are lots of examples of plastics that are bio-based that are now part of our industrial ecosystem. So it's just a matter of time.
So I'm a designer, I use materials. I'm curious to know if the manufacturer of Alginet will also be environmentally safe. The material sounds great, but will the production also be safe?
In short, yes. So one of the things that I get really excited about in creating this material is everything that goes into it. You've probably already eaten. You probably eat on a regular basis. It's biopolymers. You find them in food, and we use So I don't know if you're familiar with like textile production, but we use a wet spinning process that historically has been kind of a dirty process. But that's due to like solvent use, right? Like acetone. The solvent we use is water. So kind of everything that goes in, everything that comes out, you can eat and be safe.
Yeah.
Can you expand on your accelerator experience a little bit? Who helped you and how did it help you?
Sure. So I guess, let's see. So that was about just under a year ago. We started an accelerator, and that was a SOSV accelerator in London called Rebel Bio. And the sort of structure, they started in Cork. There's kind of a sister program out in California and called Indie Bio, and they're actually coming to New York very soon. But the whole structure there, it's 4 months, every day, and they give you an office space and a lab space. And I think, from sort of a scientific point of view, like that allowed us, like just getting access to that sort of modular built out lab space that had some of the things we needed already in it. So we weren't kind of setting it up from scratch. That was a huge advantage to the company and really propelled us very quickly, along with all the sort of business mentorship and everything that you'd expect from a sort of standard accelerator.
Yeah, so I think, when we imagine a pipeline, Biodesign Challenge starts at the beginning of the pipeline. The next step would be an accelerator program, potentially, or something along those lines, and then obviously investment. So I think what we're doing is we're building the front end of this pipeline. Hopefully these companies will stay in New York as the rest of the pipeline gets made.
What's the deal that's made between you and the accelerator for that core period, that 4 months or 6 months? I mean, how do they make the space available to you? Is it made available to you because they're just trying to foster that? It's government sponsored. Is it a private enterprise? What's the mechanic? Sure.
So I mean, all of them are different, but in kind of what I've been exposed to, it's all private enterprise. There's, this is, a venture capital firm, and what they do is invest in very early stage companies and give them the space, the mentorship to sort of be successful at an early stage.
So their investment is really in that form, and they take whatever interest in your company based on providing that.
Right. So like typical is like 5 to 10% on a convertible note. Yeah.
Okay, well, thank you, everyone. It was great to be up here.
Thank you. Thank you.
Good morning. Our next speaker is Nicole Mcknight, a PhD. Nicole is the Managing Director of Biolabs in New York. She runs the premier biotech co-working lab space at NYU Langone. Nicole is a co-founder of Keystone for incubating innovative in life sciences network, KILM, a non-for-profit accelerator for entrepreneurial scientists. A former molecular and cell biologist, Nicole performed her postdoctorate research at Mount Sinai and PhD work at the Cancer Research in the UK in the London Research Institute. She's a dual citizen, United States and England, grew up in Massachusetts and attended the University of Chicago. Welcome, Nicole.
Hi, everyone. David, Ashish, why don't you come and sit up here and, yeah, we'll do this together. So, I'm from Massachusetts. I'm a Red Sox fan and a Mets fan. I moved here from London to do my postdoc, and I had the amazing experience of growing up in Massachusetts and working in Kendall Square. And I worked at Millennium Pharmaceuticals for a summer internship and a summer job at a startup at One Kendall, and then another startup on Sydney Street. And I really experienced how exciting and helpful it is to be in an atmosphere where there are other startups, and the biotech industry is really supported by the city. And I came to New York to do my postdoc for personal reasons. My now husband works at Viacom down the street, and had this amazing opportunity to do this postdoc in neuroscience at Mount Sinai with subsidized housing, and which is important in New York because postdocs make NIH minimum, and that's the same if you live in Kansas or Boston or New York. So I came ready to either commercialize something that I invented in the lab or work at a startup company after my postdoc. And 7.5 years ago, realized, oh no, there are no startups really here in New York. What am I gonna do? This sucks. And started hanging out with all of you guys, and figured out pretty quickly that the problem was a lack of wet lab space, that there was no commercial lab space like there was in Kendall Square for companies to exist. And I was part of a group called NYC Tech Connect and started networking and hanging out with all of the other scientists who wanted to try and commercialize their inventions, and really formed KILM in order to build an affordable wet lab incubator in East Harlem. It was a grad school class on entrepreneurship that me and 4 other postdocs crashed. And so started tackling this problem of how to build an incubator, and fast forward a couple of years, and 4 years ago I teamed up with a group called Biolabs, which started in Kendall Square and built Lab Central. So they developed this model of how biotech companies can actually work together. You would think, because their intellectual property and their inventions are their assets, and that they would want to be secretive and protect it, but actually it's the opposite. When you get these scientists working together in a collaborative place, they learn so much from each other, and they share, and they're able to really get going. So what do we provide at Biolabs? It's actual infrastructure. We have 50,000 square feet of space in Hudson Square on Varick Street in the old ImClone labs. And it's half wet lab, half office. So the office space is private offices, desks for the scientists, and conference rooms. And then on the lab floor, it's actual science wet lab benches. The scientists rent a bench, it's a 5-foot bench, and with that, everything included, they get access to a cold room, a tissue culture suite, a freezer farm, a microscope, all this equipment that we get, so that really they can raise a little bit of money. They come out of accelerators, they raise some money from friends and family, and they're ready to go. And they can move in and they can start doing their experiments and really accelerate how quickly they can raise more money. So we can house those one scientists with an invention spun out of their academic institution, something like coming out of Bridge Medicines or any of the other really great things that are coming out of our amazing academic institutions. Or we can have companies that are a bit bigger and have been able to raise a decent amount, millions of dollars from venture capital companies and move straight into one of our private lab suites. 4 years ago, I was pounding the pavement, actually with Bill, looking for 30,000 square feet of space that would support us, manufacturing zone space that would support the infrastructure. In 2015, we raised $2 million from the state through Empire State Development. And in 2017, we answered the first RFP that the city put out, the NYC EDC put out through their $500 million LifeSite NYC initiative. So $7 million supported in partnership with NYU Langone, who have been an awesome, awesome partner. And we are just completing the renovations on this 50,000 square feet of space. Those who know me, you know it's been a roller coaster, and it's been a long time coming, and construction in New York is crazy. But we're there, and I want to thank. So JB&B is here. There are engineers. Gensler is our architect, and Skanska is our construction managers, and it's been a really, really great team. And also NYU, they've been great. And we are supported, Biolabs at NYU Langone is supported by sponsors. So Big Pharma, Bristol Myers Squibb is one of our founding sponsors, Eli Lilly, Sanofi, Boehringer Ingelheim. They actually give cash to support our operations, so we pass those subsidies down. We also have great sponsors who provide all the equipment. Even Herman Miller is our new sponsor as well, donating furniture. So it's really a big team effort, and it's all so that we can pass on this infrastructure to our resident companies who need it so badly because they're just starting out. So a year ago, we opened a 1,500 square foot interim space, a little baby co-working suite on the 5th floor at 180 Varick Street. And these guys are resident companies in that space. And there's something really exciting about New York whereby the tech industry has done so well here, and they haven't needed the wet lab infrastructure and the lab space. And it's really taken off, but there are these companies that are in the intersection of biology, life sciences, tech, who we've been really enjoying being able to help without the wet lab space. So David and Ashish are going to talk about their startup companies. These guys will always be my favorites because the first cohort of companies. But it's really exciting, and it really shows the future of and the potential here in New York City for these really innovative companies that are at the intersection of fashion and bioprocessing and tech and big data and AI. And then we'll have these therapeutics companies coming out too. So we're really excited to be able to house them all and support them and really showcase these companies in our gorgeous new space. So yeah, I'm going to hand it to David here.
Okay, thank you.
Yeah.
Thank you, Nicole. My name is David Rocklin. So I am the co-founder and CEO of Redesign Science. We were one of the first companies into this new biolab space, taking advantage of the fact that we are, what we refer to as an in silico pharmaceutical company. Everything we do within the company is on the computers, but we develop partnerships in therapeutics development. And that depends on experimental work. And as the resources expand and as our projects develop, we may begin to do some of that wet work ourselves. But at this point, we're very lean. And the basis of this company is technology that I started tinkering with at NYU as a graduate student and a postdoc. And very early on saw the potential for commercial potential, for what I was doing. And that started about 4 or 5 years ago, early developing the technology, but the company itself is just about a year old. And what we do is we're building and applying a computational platform with the goal of speeding up new drug discovery against new drug targets. And we focus on what we refer to as the lead discovery stage. Now, this is really like the ideation stage for new drug development, and we do that through atomic level simulations of the drug targets themselves to identify new opportunities for druggability. So over the last 10 years, fields that we refer to as the omics space, this is like genomics, transcriptomics, proteomics, and I'll put artificial intelligence and machine learning, in there as well. The convergence of these new technologies is leading to a much deeper understanding of the underpinnings of disease biology. And this is leading to sort of a wave of emerging drug target classes. And these are sort of in depictions of some of these new drug target classes that are emerging from these new technologies. Now, what makes these interesting is that they largely lay outside the wheelhouse of traditional pharmaceutical drug discovery programs, and they remain very sort of challenging, just out of the reach of many of the conventional drug development technologies. The reason it actually has to do with the biomolecular structure of these targets. They're much larger, or they're dynamic, and they have shapes that scientists aren't accustomed to designing molecules against. So it requires a lot of innovation. The way we approach this with our in silico platform is by actually running atomic level simulations of the drug targets themselves, as you can see here. And then we develop technology that essentially listens to the surface of these drug targets and processes large amounts of data from these simulations to identify specific opportunities for druggability. And then we go in and represent these protein surfaces, the shapes of these protein surfaces, and we use these shapes to directly guide the development of new small molecules. So our business model is very partnership-centric. So we sit here right in the middle of this lead discovery stage, and we look to form partnerships both upstream with target discovery companies. Now, these can be, biology labs with great expertise in specific diseases, but more and more these are becoming artificial intelligence companies and machine learning-based companies. And then we also partner downstream in what is called the lead optimization stage. This is taking these ideas for new drugs and really tinkering with them to get them into a therapeutic, a viable and non-toxic therapeutic. And these companies, there are also many artificial intelligence companies and methodologies called adaptive learning. So our vision is to sort of create a technology sort of bridges these two, and we're very interested in creating a completely in silico pipeline to accelerate new drug discovery process by 1 to 2 orders of magnitude. So that's 10 to 100 times faster. And the goal is to form partnerships around new therapeutic ideas, develop these new therapeutic ideas to the point where we can then partner with pharmaceutical companies to help us co-develop these into the clinical trial stage. So we have 3 new drug programs already in development. And one of our lead asset is actually already a partnership that was established since we've been at Biolabs, a new company that we're forming with one of the biologists that we met through being in this co-working space. And we're looking forward to forming more of these partnerships over the next 5 years, and helping to stimulate this growing ecosystem in New York City.
Great, so I'm going to introduce Ashish here. Sorry, I haven't left you too much time, but.
Okay. I'll try to go quick. All right, hi everyone. My name is Ashish Agarwal. I'm the founder of SolveView. We're building a data science platform for working with life sciences data, more specifically genomics data as our primary focus. So I could go to the next slide. Oh, I do it myself, right? Okay, so there we go. Okay, so the problem we're trying to address is basically that genomics, like many other industries, is exploding with data. The data is very complicated. Just as a quick example of the current state of things, I was on a project once at a great institution that you all know with fine people, where they spent $800,000 generating sequencing data, and I was tasked with finding the data. They simply did not know what computer it was on 3 years after the data was generated. So that's sort of an extreme example, but the situation is more fundamental, and it's not that easy to solve. The data is very complicated, and people simply don't know how to manage it. Dealing with the algorithms in this space is also intensely complicated. Genomics specifically is an industry that deals with thousands of algorithms. For any single analysis, you need tens of algorithms. The algorithms are all free, okay? But just because somebody gave you the code doesn't mean you can run it, okay? So this is a myth about programming, about code, that if you have the code, you can do something with it. That's actually largely false, okay? People can give you code, and then you're stuck, and you have no idea how to run it, okay? So what is the solution? What do we need? We need to have a data management system that does not exist in this industry currently. Everybody's building their own system, and that's just how it is right now. Once you have the data managed, you need to be able to analyze it. The algorithms are very complicated, they're diverse, there's no API standards, there's no uniformity, and they're very computationally intensive to run. Unfortunately, these algorithms are very complicated. Another myth is that when you run an algorithm, you get an answer out. That's not really true. You run an algorithm, and you get a bunch more data out, okay? And you have more mess, okay? So what you really need is the ability to explore that data, the results, in some intuitive and useful way. So the SolveView platform is trying to address this problem holistically. We handle all the infrastructure. We provide a data management system. We enable you to integrate with all the data sets that you want publicly, but of course, upload your own data sets and keep that very private and secure. We then make it possible for you to run all the different algorithms you want, and we have a wonderful user interface where we allow you to visualize the data sets and explore it interactively. So as you can see, we are a system that's trying to be very holistic and end-to-end. We're not based on like cracking a single problem. We didn't come up with one invention and build a company on that. We really have a whole suite of patents that we're planning to apply for, and we're trying to solve this problem holistically. That's both required for technical reasons, and we also think it's a good business strategy. It's what the industry needs. Nonetheless, we do have some focus. We're not trying to do it all at once. So initially, microbiome, cancer, and agriculture are some of our focus areas, and that's just driven by our early customer relationships, so that we know people in those spaces. But our technology is really more general. So thanks to my whole team. Raj is our director of genomics. We have some great advisors. Tom Cerrito, who is the founder of Crosswalk Ventures. He's helping build a venture fund for life science companies in New York City. Nick Sochi is the director of bioinformatics at Sloan Kettering. And Bob Harper is one of the great minds of computer science that gives us guidance on the technical aspects of our work. Okay.
Great. So, it's exciting times. You've seen 3 great presentations from 3 really exciting companies. And I want to thank Nancy for organizing and the team here for a great inaugural event, and really happy to be able to present to everybody and share our enthusiasm and excitement for this next chapter of biotech in New York City.